Let me begin with this one. That is what I have selected a topic for today's we call discussion there in this virtual Zoom meeting of organized by Magnus Group. That is understanding drug transport in plasma and the role of protein binding in particular. As has been introduced by Dr. Deepika, I am from the Department of Pharmaceutical Chemistry of the Faculty of Pharmaceutical Sciences, UCSI University, Kuala Lumpur, Malaysia. And before I should go for in detail about the work there, I will give you a little bit of introduction actually. That is to the drug protein binding. The interaction between drugs and plasma proteins, the process is usually reversible. And the importance of this work is because it can affect the drug distribution, efficacy, and elimination. Major site of the drug protein binding is blood plasma. Now, the key plasma proteins which are involved in this interaction, here we can have got plasma albumin. And then what is important for this plasma albumin, because it binds acidic as well as neutral drugs. Once I say acidic drugs, it means the drugs have got acidic group, particularly the carboxyl group. On the other hand, the basic drugs which are having amino group there, maybe the first primary or secondary amino group, just like we call phenacetin acetaminophen, alpha-1 acid glycoprotein, which is available in plasma, will be able to bind the basic drug. On the other hand, lipoproteins and globulins, they are less commonly involved, so that whenever you are talking about the proteins in plasma, these are the two main proteins actually, plasma albumin playing the major role, again followed by alpha-1 acid glycoprotein. Now, whenever we talk about the interaction between the drugs and proteins, so that people talk about binding characteristics. So here, little bit idea about that is drugs are going to bind to the protein, and this binding is reversible, and then it is involving the non-covalent interactions. Non-covalent interactions, as you might be aware of, involving hydrogen bonds, electrostatic interactions, hydrophobic interactions, van der Waals forces, so on and so forth. Now, this binding may be saturable, means that the binding sites may be fully occupied, or the binding may be selective, that some drugs prefer specific proteins to bind. And this is also influenced by the drug concentrations and their affinity towards the proteins. In other words, when the drug is going to bind to protein and the binding is not saturable, then you can have that the drug is existing in the two forms. The one is the free or unbound form, and the other one is the bound form. And then whenever you are talking about the activity, pharmacological activity, it is the free or unbound form which is showing the pharmacological action. On the other hand, the bound drug, it acts as a reservoir, but it is inactive actually. And then remember only that the free drug, which can cross the membrane actually, and then it can bind to the site of the drug action, that is where the receptors are available. Or another fate will be that the free drug can be metabolized and excreted. And then whenever we are talking about the binding affinity and capacity, these are the common words that people are using whenever you are talking about a drug-protein interaction, so that you'll be having some drugs are called high affinity drugs. They bind tightly, but they bind less. Or high capacity sites where they can bind more drugs with a lower affinity. And whenever you are talking about the binding, it is basically described by association constant or the dissociation constant, which is Ka and Kd. Factors which can affect the binding process, that is number one, is the level of the proteins in blood plasma. Because in certain disease states, for example, liver and kidney diseases, the level of this albumin as well as alpha-1 acid glycoprotein that may increase or decrease there. Secondly, you may call the drug concentration. And thirdly, you will be having presence of other drugs, which is very important here, because several other drugs, they can compete actually with each other, so that you can have got the drug-drug interaction. And then the pH and temperature. Regarding the clinical significance of the drug-protein interaction, here we will talk about drug efficacy. On the right side, probably you can see that is only the free drug acts on the receptors there. The one drug can be differentiated from other drug in terms of efficacy as well as in terms of the half-life there. Half-life, the bound drugs prolong the duration. That's what I say the advantage. Although the bound drug is not going to show the pharmacological response, but then it can increase the half-life so that prolong the duration. of stay of the drug in the blood circulation. Now, drug interactions, as I told you just now, that there is a competition for the binding sites there, so that the two drugs can compete for the same binding sites on the protein molecule. And then, in fact, in hypoalbuminemia, as just now I was talking about, that is in the disease states when the concentration of protein is either increased or decreased, you may have got the drug-protein interaction is also playing some other role. Therefore, when you say the drug-drug interaction, or you can say displacement interaction, means one drug displaces another from the protein-bound form here, and therefore it can increase the free concentration of the displaced drug. Means the drug which was already bound with the protein molecule, and then when you are taking another drug, when this drug is having higher affinity, it will be able to displace, and provided that it is going to bind to the same site, it can displace, you may call the drug, and therefore the free concentration of the free drug of the first drug actually that will increase, and then it can be excreted there. Examples are warfarin and sulfonamides. And therefore, once the free drug is available there, you may have got it may lead to the toxicity. The cartoon you can have got here, this is the structure of albumin, where you can have got, you may call that albumin is having, you may call the two sites, main sites actually for the drug binding. And suppose if the drug is sitting somewhere here on site 1, then it can be, we call, displaced by another drug number 2, which is going to bind to the same site with a higher affinity. Now, this is also I talked about just now, that changes in the disease states. For example, in liver disease, albumin concentration goes down, and then once albumin concentration is going down, the concentration of the free drug will increase here. Similarly, in kidney disease, uremia, you will be having altered protein binding, or inflammation, you will be having the alpha-1 acid glycoprotein will increase. Therefore, the changes in the concentration of the proteins in blood plasma that will affect, you may call, the protein binding of the drug molecule. Therefore, the ratio of the bound and the free drug that will be affected there. And it will have got the implications in the drug dosing. For example, highly bound drugs, a smaller volume of distribution, dose adjustment have to be made in low protein states, and monitoring of the free drug levels under the critical situation or in critical care. Here's examples of some highly bound drugs: warfarin, 99% bound; phenytoin, 90% approximately, 90% bound; diazepam, propranolol, naproxen, the high binding drugs, and therefore for these drugs, chemical monitoring is needed there. Laboratory measurements for determining, you may call, the concentrations of the free and the bound drugs actually. So these are the two main techniques which are used: ultrafiltration and equilibrium dialysis, to separate, you may call, the free and the bound drugs. Protein binding studies during the drug development are needed there, and interpretation of free versus total drug levels are needed there. Recent advances and challenges in this particular topic there, that protein binding in special population, particularly neonates, or the use of modeling, that is the PBPK, that is physiologically based pharmacokinetic data in drug development, and role in personalized medicine. Now, I can take you, may call, a little bit tour of, you may call, the two drugs particularly, which we have selected here. But before that one, again, I can refresh your memory about the drug administration and pharmacokinetics here. If you look over this cartoon on the left side, that is, if you look over, that is, the drug can enter into the body through different routes: parenteral, that is intravenous, muscular, or subcutaneous, through the injection, or you can have got sublingual, or through inhalation, or oral, or transdermal patch, or the topical. And then the drug is reaching to the circulation. And then once you talk about the circulation, you will be having, you may call this one, that is pharmacokinetic studies, where we say, you may call ADMA, that is absorption, distribution, metabolism, and excretion. Here, you may call, the talk is confined towards the distribution. That is how the drug is being distributed. And for the distribution, as I told you, that is, it is going via, you may call, the blood circulation, where the drugs are going to bind to the protein molecules. That's why the drug-protein interaction we are interested in. Here you can have got again another cartoon to show you, may call, the injection of the drug when it's reaching to the blood circulation, and then via blood circulation it is reaching to the target organ. And then what we want here, that is, in the blood circulation, you will be having blood is being distributed into two forms. One is, you may call, which is bound to the protein. So here can have got the green dots are showing about the free drug molecules, and then the blue dots are the proteins, so the protein bound drugs. So that is the distribution. And remember, it is the free drug which is going to show the pharmacological response. The bound drug can prolong, you may call, the stay of the drug, that is the half-life. So importance of the drug plasma protein interaction, again, that is what we say, we call these interactions help to control the distribution of the drug, rate of excretion of the drug from the body, toxicity, increasing the drug solubility and increasing the drug's half-life. And here, this is another cartoon where you can have got in plasma what is happening, that the drug is interacting with the protein, forming drug protein complex, and then the free drug as well as the bound drug, they are in equilibrium. Now, free drug is also reaching, you may call, to the liver actually, and then by the action of enzymes, it can be metabolized and excreted into the urine and bile. This drug can also reach to the kidneys there through the carrier excretion into the urine, or the drug can reach, you may call, to the target site there, where it can bind to the receptor, forming the drug receptor complex, and then you can have got the pharmacological response. As I told you, the competition between the two drugs or the compounds for the same protein, so that may lead to the drug-drug interaction and drug displacement. Assessment of the drug protein binding in blood is important part of the ADME data actually, and needed prior to the approval of any new pharmaceutical agent. Here I will give you the, I may call the partial work of one of the PhD student, that is interaction study between hepatitis B and C virus inhibitors and human serum albumin. That is here, hepatitis B and C diseases are contagious liver diseases, infection with HBV and HCV. Infection causes both acute as well as chronic liver disease. Global health problem, because 250 million individuals are chronically infected with HBV. This is a little bit old data, 2020. 71.1 million individuals are chronically infected with HCV, also the little bit old data, 2021. These are the drugs which have been being used actually. These are the purine and pyrimidine analogs of, you may call, the one is a PSI 6206, or you can also get this one another name, that is RO2433 PSI. This is a uridine analog. This is a drug used for the treatment of HCV, prevents RNA synthesis by inhibiting RNA-directed RNA polymerase. And then another drug which has been selected here, this is entecavir. It is a guanidine analog there. That is what we call ETR in short. This is the drug used for the treatment of HBV. It is also preventing, you may call, viral application by inhibiting all three enzymes used in HBV polymerase. This is the main, you may call, the protein through which, you may call, we have studied the binding there, that is human serum albumin. I will give you a little bit details about it. That's the most abundant protein in circulation. It is a major transport protein, single chain polypeptide, 585 amino acid residues. It is having, you may call, three homologous domains, 1, 2, 3. That's what you can see, you may call, this is a red color one, blue one is 2, and the green one is 3. Each domain is divided into subdomains, 1A, 1B, 2A, 2B, 3A, 3B. And then there are three well-known binding sites actually available there. And that is, we say site 1, site 2, and site 3. Site 1 is also known as Sudlos site 1. Site 2 is Sudlos site 2, and site 3 is, this site 1 is located in subdomain 2A. Site 2 is located in subdomain 3A, and site 3 is located in subdomain 1B. With this background, this is another picture where you can see, you may call, that is again the same structure, crystal structure of, you may call, the human serum albumin. And these are the three sites where the different drugs are going to bind to site 1, site 2, and site 3. The methodology which has been used to study here, that we have done, you may call, computational methods as well as, you may call, the wet lab experiments. Computational methods is usually the molecular docking using Autodock tools, where we have done the cluster analysis at site 1 and site 2 primarily there, because we have not done the site 3. And then we have done the binding orientation analysis and leak plot plus analysis to see the interactions, followed by molecular dynamic simulation, MD simulation, to find out total energy, RMSF, RMSD, and distance plots, because to check, you may call, the stability of the drug protein complex. Whatever the information we get from the computational methods actually, that is, we want to verify through the wet lab experiments. That's what we say, experimental methods, where we have done the incubation of the drug, either PSI or the ETR with HSA for a certain period of time to attain the binding equilibrium. And then we have used, you may call, the different techniques actually, usually fluorescence, absorption, and the CD spectroscopy, supported by, you may call, the atomic force microscopy. These are the wet lab experiments to find out, you may call, the quenching mechanism and binding properties, binding thermodynamics, intermolecular forces, conformation studies, and the displacement studies. Here you can have got the results of, you may call, the docking analysis for the PSI, because this, remember, this is the drug for the HCV, and then PCI HSA system. We have confined only for site 1 and site 2. Here you can see the cluster analysis where the number of conformation against the mean binding energy. Remember that at the particular site, you may call, the site has been shown with the, you may call, the red one is site 1, blue one is site 2. The clusters which are having maximum negative binding energy, it means they are giving rise to, we call, the more stable complex formation. So cluster analysis of 100 docking runs of PSI to site 1 and site 2 in the HSA crystal structure 1BM0. And then here you can see probably site 1 is having, we call, more clusters with a greater negative energy. It means compared to site 2, site 1 is more preferred. We have done the orientation analysis to find out, you may call, and then the Lick plot plus analysis to find out the hydrogen bonds formed as well as the surrounding hydrophobic residues. Based on this information, we reached to the conclusion that the binding preference for this PSI is, you may call, for site 1. Then we have done the MD simulation studies there for the PSI HSA system. Here you can have got the total energy of, you may call, the system. This is RMSF plot, this is RMSD, and the distance. You can have got, you may call, here the final conclusion, that is average total energy of minus 1.52 10 power 6 kilojoule per mole. Then RMSF showed, you may call, the normal flexibility. RMSD value for stable PSI HSA complex, that is a green one. And then the distance between the PSI HSA remained consistent, less than 0.3 nanometer in MD simulation studies, suggesting, you may call, that the PSI HSA complex at site 1 is, you may call, stable. Similarly, we have done the docking analysis for the second drug, that is ETR HSA system. Again, this is for site 1 and site 2. Site 1 shown with the red color cluster analysis, site 2 with the blue color. Again, you can see, you may call, at site 1, the number of conformations more with a higher negative energy, negative value of mean binding energy, suggesting, you may call, that site 1 is the preferred site over than that of site 2. And here, this is the orientation analysis and the Lick plot analysis, which is suggesting, you may call, the hydrogen bonds which are formed, particularly here in this case, 4 hydrogen bonds are formed between the drug and the protein molecule, supported by, you may call, the hydrophobic residues, hydrophobic interaction. In the previous one, you have seen, you may call, that is the 6 hydrogen bonds are being formed there in the Lick plot plus analysis. This is the MD simulation analysis of ETR HSA system. You will be having here, that is average total energy of minus 1.521, and the system is stable. RMSF showed the normal flexibility. RMSD value inferred is stable. The ETR HSA complex also, and the distance in the last one between the ETR HSA remained consistent, less than 0.2 nanometer in MD simulations. Then, after getting, you may call, this information from the computational methods, actually, that is getting the information that site 1 is the involved and which is the preferred site, and then the drug is going to bind to albumin there. We have done, you may call, wet lab experiments using fluorescence spectroscopy. Here, this is the fluorescence quenching data, that is the titration data of, you may call, the PSI on the left side, ETR on the right hand side. You can see the top spectrum is the spectrum of human serum albumin fluorescence spectrum after excitation at 280 nanometer. And then once you are titrating this protein with increasing concentration of the drug, you can see, you may call, fluorescence intensity decreases, that is the quenching is taking place. Same thing happens, you may call, the quenching is taking place here. The quenching is of the protein fluorescence is an indicator. That's what we say. Quenching in the fluorescence spectral signal is the indication of the interaction between the PSI or the ETR and HSA, suggesting, you may call, the complex formation. Then this fluorescence quenching data were analyzed using the Stern-Warmer plots, using the equation, Stern-Warmer equation. These are the plots which have been shown. And now these fluorescence quenching studies have been performed at 3 different temperatures, so that each titration has been performed, you may call it minimum 3 different replicates. And then at 3 different temperatures, we have got, you may call, the fluorescence quenching titration transform the data using Stern-Warmer equation, we got the Stern-Warmer plots. So these are the Stern-Warmer plots at 3 different temperatures, 290, 300, and 310K. On the right side, also the same temperatures, the fluorescence quenching titration with ETR. The left one is PSI. And then here you can see that is all the plots are linear. So linear plots are suggesting involvement of one quenching mechanism. Because if you talk about the quenching of the protein fluorescence, it can be the collisional quenching, it can be the static quenching, or it can be the mixture of two. So you will be having the phenomenon that is both the quenching mechanism are involved in certain direct binding direct protein interaction, whereas you'll be having that is only one type of quenching will be involved, that is either the collisional or the static there. So very first information you can get it there from the Stern-Warmer plots. If the Stern-Warmer plots are linear, it means they are suggesting that the only one type of quenching mechanism is involved there. Now here I will give you a little bit idea about this is the Stern-Warmer equation. There will be having KSV is the Stern-Warmer constant, KQ is the bimolecular quenching rate constant, and then you will be having you should remember this thing, that is the correlation between the KSV and with temperature. Suppose upon increasing the temperature, if the value of the KSV is increasing, it reflects that the collisional quenching is involved. Collisional or the dynamic quenching is involved. On the other hand, if the value of the KSV decreases there upon increasing the temperature, it means it is the indicator of static quenching is involved. Or you can say when the static quenching is involved, it means ligand protein complex formation. So you have already seen that the linear plots have been observed and which are showing involvement of one quenching mechanism. And then we have to decide if you ask me, that is why we have carried out the titration at 3 different temperatures, minimum 3 different temperatures. Usually people have to do 3 or maybe 4 different temperatures to find out this correlation, whether the KSV value is increasing or decreasing. And here you'll be having the data. That is, in this table, you'll be having the left one is the PSI HSA system. On the right side is the ETR HSA system. These are the temperatures and the KSV value in the middle column, and then the KQ value, bimolecular quenching rate constant. So here can have that. If you'll be having the temperature is increasing, you can see the KSV is decreasing there from 6.03 to 4.93 into 10 power 3. Same in ETR HSA system also, from 2.12 to 1.39. Also, the value of the KSV is decreasing. It means you may call that is an inverse correlation between the temperature and KSV. And as I showed you in the previous slide, that if the correlation is inverse, you may call between the temperature and KSV, we can say the type of quenching involved is static quenching. That's what we say here. There is another parameter, that's what we say KQ parameter, bimolecular quenching rate constant. If the value of bimolecular quenching rate constant is more than 10 power 10, this is another indicator of the static quenching. So here you can have the value of the KQ is whatever the numbers here multiplied by 10 power 12, which is higher than 10 power 10 per molar per second. That is another indicator that is a mechanism of quenching is static quenching. Once you say static quenching, it means it is a complex formation. In order to further support that is the static quenching is involved or the complex formation is taking place, we have done absorption spectral analysis actually. So here can have that is the absorption spectra of the protein alone, and then titrating protein with increasing concentration of the drug. The bottom spectra is the spectra of the protein alone actually, which you can see if my cursor is there. And then when you are adding the drug into the protein solution, you will be having absorption spectrum is increasing there. That's what in both cases increase in the absorption spectrum or increase in the absorbance value. The absorption spectrum of the free drug has been shown here, A, and here A, that is for the PSI. This one is ETR. And then since you'll be having the protein absorption is usually the absorption maximized around 280 nanometer, because remember HSA is considered as having only single tryptophan residues. Although it is having 585 amino acid residue, but the tryptophan is single. So when you are having the absorption maximized 280 is the indicator of the tryptophan. And then you see if you are adding the drug, you will be having the increase in the absorbance values. So here this plot you will be having, this is the HSA, PSI, complex once you are adding it, so that you can see chemical absorbance is increasing. And the bottom plot is you may call the summation of the absorbance value of HSA as well as absorbance value of the PSI. And then you see we call still there is a difference between the two. In both cases, that's what I say, increased protein absorption upon added PSI or ETR. You'll be having a steady quenching, and that is suggesting you may call this is not CLV. It is you may call, you can say that is PSI or ETR HSA complex formation. Then we have used the same the quenching data actually to transform them into the double log plots. So these are the double log plots of using we call against we call these log F0 minus F upon F. This is F0 is the fluorescence intensity in the absence of the drug. F is we call in the presence of drug, and three different temperatures. From these double log plots, you can get we call the value of the association constant or the binding constant, or you say binding affinity. Here you'll be having we call the binding affinity of this one. This is the PSI. Here this one are for ETR. You'll find we call the binding affinity has been found in the range of 10 power 3 or 10 power 4. That suggests we call the binding affinity is moderate between we call the drug and the protein. Remember that binding affinity, once you talk about, it means it can be classified into strong binding affinity, moderate binding affinity, and low binding affinity. And if you ask about, you may call that which affinity is required, actually the answer is moderate affinity is required, because if the binding affinity is very high, you may call that is very strong, then the transport protein which is carrying the drug may not be able to deliver the drug at the target site there. So drug will not be released. On the other hand, if the binding affinity is very small, you may call, then again the drug may not be carried by the protein molecule. So here you can have what you may call moderate binding affinity based on these Ka values. Values of N is number of binding sites, is suggesting you may call that is close to 1. It is suggesting you may call single binding site is available in the protein molecule for these. And then you can have got another thing, that is the Ka value is decreasing there upon increasing the temperature. And that is suggesting actually that once you increase the temperature, K is decreasing. It means it is an exothermic reaction. So in both cases, you may call it is decreasing. So then both the reactions, the protein drug binding reaction, exothermic. We have also done the one-half plot actually using this data there at 3 different temperature to get the thermodynamic parameters there, entropy change, enthalpy, and free energy change. You can see here now that is the free energy change is negative actually, which is suggesting you may call the process of binding process is continuous process. On the other hand, you will be having here the delta H values also negative for both systems. That is showing you may call exothermic reaction. And then regarding the forces involved, that you can get the information from here. That is here you can have that because that if the delta H is negative, we can predict because that van der Waals forces or hydrogen bonds are involved. On the other hand, if the delta H is positive, we can say hydrophobic interactions or binding site dissolution that is taking place there. So this is the structure of the PSI. You can see the different groups which can be involved in the hydrogen bonding there. And then you'll be having this is the carbon skeleton, which can also involve there in binding through the hydrophobic interactions. We have also done microenvironmental alterations here. So microenvironmental changes. So these are the 3D fluorescence spectra of the free protein, that albumin, and then the drug protein complex there. Now you can see on the right hand side, this is the data which is available, the peak position. Particularly, you have to concentrate over here that the peak number 1 and 2. Peak 1 and 2 is due to the protein, tryptophan and tyrosine residues. Protein and the peaks A and B, these are the scattering peaks actually. So what happens once you are adding the drug into the system, you will be having the both peaks are going to be the fluorescence intensity of both peaks is going to decrease. That is suggesting that the microenvironment around the tryptophan and tyrosine residues is affected there upon the drug binding. Here you'll be having the same microenvironmental alteration, the 3D fluorescence spectral data of HSA, and then we call it the ETR. So this is the left side, this is the protein alone, and this is upon addition of the drug. You can see we call here again, that is the peak 1 and peak 2, the fluorescence intensity that is changing 36 and 89%, suggesting the microenvironment around tyrosine and tryptophan that is being affected there. So decrease in fluorescence intensity, micronomental alterations in HSA around tyrosine and tryptophan residues. We have also checked in whether is there any secondary and tertiary structural changes in HSA molecule using CD spectroscopy. This one left side is the far UV CD spectra, which will tell you about the secondary structural changes. On the right side is the near UV CD spectra, which is telling about the tertiary structural changes. Here you can have this is the helical protein, because the two minima you will be having at 208 to 222 nanometer. Solid line is the spectra for protein alone. Dash line is with the drug. You'll be having a slight rise in the spectral signals of both far UV and near UV CD spectra, showed mild changes in HSA secondary and tertiary structures in both cases actually. Similarly, here can have the similar kind of picture, that is upon addition of the drug, you'll be having slight changes in the spectra, both in the far UV and near UV region. We have also checked the morphological changes through atomic force microscopy. So you can see here on the left side, you will be having the protein alone, and when the drug has been added, you'll be having the protein structure morphology is being changed. That's what we say, because protein molecules become swollen upon PSI or ATR addition. Therefore, that will also give you the indication that PSI, ATR, HSA complex formation that is taking place. Then lastly, we have done the binding site of characterization, because the computational methods have suggested us that it is the site 1 which is involved. So we wanted to verify using the wet lab experiment, that is a competitive drug displacement experiment using the fluorescence spectroscopy. We have used the site markers actually, site 1 and site 2 markers actually. They are the well-known markers. These are the drug molecules which are well known to bind to site 1 or site 2. Warfarin, indomethacin, well-known site markers for site 1 markers, and diazepam, ketoprofen is site 2 marker. Here you can have that if you look over this one, the top fluorescence spectrum of warfarin protein complex here. So it means the warfarin is already bound to the protein. Now, in that complex, once you add the drug, the PSI, you will be having that there is a quenching, the fluorescence intensity is decreasing. So since this is the signal of the warfarin HSA complex, this signal is decreasing, suggesting that probably warfarin is being displaced, and that's what you can see the displacement over here. Here you can have once we talk about the quenching of the protein alone, and then protein when it is bound with indomethacin, which is also binding to site 1, we found the decrease in the fluorescence intensity is different in both the system, again supporting the warfarin results, suggesting site 1 is the preferred site. On the other hand, when we have used the diazepam, so that is the diazepam HSA complex. Diazepam is the site marker for the site 2 actually. And then you can see here that the decrease in the fluorescence intensity in both cases is the same. And that shows conclusion that the PSI binds to site 1 and then non-involvement of site 2, just confirming the computational data. Same thing we have done also this competitive drug displacement for ETR. So here again, the same thing, that is warfarin HSA complex, and then upon addition of the drug, we will be having the decrease in the intensity. So suggesting warfarin is being displaced, it means the drug is going to bind to the site 1 actually. And here also phenylbutazone, which is also the site 1 marker. So you see this one is the red one is with the protein alone, and the blue one is the PBZ HSA complex. Again, you'll be having the difference, so that you can have that that probably site 1 supporting the warfarin data, that site 1 is the preferred site. On the other hand, ketoprofen site 2 marker, here you can see whether you take protein alone or the KT and HSA mixture, you'll be having the same change in the fluorescence intensity, suggesting ETR also bind to site 1 and non-involvement of site 2. So in conclusion, we say PSA, PSI, ETR showed favorable interaction with HSA. Quenching of the protein fluorescence by PSI and ETR was characterized as a static quenching. Changes in protein conformation upon ligand binding were observed. Ligand protein complexes were stabilized by van der Waals forces, hydrophobic interactions, and hydrogen bonds. PSI and ETR exhibited a clear preference for pseudo site 1, that is located in subdomain 2A. These are some of the references which have been cited there in some of the slides actually. And these two, the binding studies were published already. This is not very new, 2023 and 2024. And this is the student actually from Nigeria, actually, that is which was working in University of Malaya there. Mujahid Abu Bakr. It is a part of his PhD studies actually. Uh, Dr. Sharuddin from University of Malaya, Associate Professor. He is my collaborator. He is from the bioinformatics group, so particularly computational work is under his supervision. Dr. Diani is the senior lecturer, also my student there. So she was also supervising UMICOL. The work has been carried out both at Institute Science Biology, Faculty Science, University of Malaya, as well as UCSI University. And then lastly, I would like to thank Simikal for your patient listening. If you are having any questions, you can ask me now.